The Presage ST2 assay system is intended for the in vitro quantitative determination of ST2 in human serum or plasma. ST2 is used as an aid in risk stratification of patients with heart failure. In this video, we'll show you everything you need to know about running the assay properly. You can review any section of this video or your package insert anytime you have questions. Let's get started. Let's begin by reviewing the components of your Presage ST2 assay kit. The assay kit comes with 2 vials of calibrator. Note that each vial is labeled with the appropriate water reconstitution volume. The kit also comes with 1 bottle of standard diluent, 1 bottle sample diluent, 1 bottle of biotinylated antibody reagent, 1 bottle of streptavidin HRP diluent, a vial of streptavidin HRP 100X stock, 1 bottle of TMB, 1 bottle of stop solution, 1 bottle of wash buffer, and 1 96-well antibody-coated plate. Now let's review the control kit. Each control kit comes with 1 vial of low control and 1 vial of high control. Note that each vial is labeled with the appropriate water reconstitution volume. Also included with each kit is an instruction for use booklet. There are laboratory materials required but not included with the Presage ST2 assay and control kits. These items are a 96-well non-binding transfer plate, a timer, micro pipettes and pipette tips, a mechanical plate washer or wash bottle, a plate shaker, a plate reader, vortex, test tubes, and reagent reservoirs. A 1-liter graduated cylinder is also needed. We begin the assay setup by allowing all components to come to room temperature. After they have come to room temperature, the first thing you will want to do is reconstitute the assay kit calibrator and the control kit. To do this, you will remove the foil top from each of the vials. It is a good idea to label the rubber caps for the calibrator and the control so that you know which cap is associated with each vial. You will also want to note the date of reconstitution so that if you need to reuse these materials, you know at which date they expire. You will note that each vial has the reconstitution volume labeled on it. It is best practice to allow the vials to rest without mixing for a minimum of 20 minutes. Next, we will prepare our streptavidin conjugate reagent. You will only want to make enough conjugate to account for the number of patient samples that you will be running. In this video, we will be running 38 patient samples, which requires an entire Presage ST2 assay kit. Therefore, we will make 13 mils of the conjugate diluent. To do this, transfer 13 mils from the conjugate diluent bottle into an appropriate size test tube. Next, we will dilute the 100X conjugate into the streptavidin conjugate reagent. It is a 1:100 dilution, so therefore we will spike in 130 microliters of the 100X. Gently invert the test tube to allow the solution to mix. It can then be placed off to the side until use at a later time. To make the wash buffer, you will need 950 milliliters of water and 1 bottle of wash buffer. Simply pour the 50 ml of wash buffer into the graduated cylinder. And cover with parafilm. It's a good idea to gently invert the graduated cylinder to allow the solution to completely mix. It is now ready for use. Next, we will prepare our patient sample dilutions. For this, we will need our transfer plate, the sample diluent, and the thawed patient samples, and a reagent reservoir. This will be a 2-step dilution process. The first step will be a 1 to 10 dilution of the patient samples, and then a further dilution to make a final 1 to 50 dilution. In the first 5 rows of the transfer plate, you will add in 180 microliters of sample diluent. In the last 5 rows of the transfer plate, you will add in 200 microliters of sample diluent. Next, we will begin by transferring the patient samples onto the transfer plate. You will want to make sure that the samples have thawed and been brought up to room temperature. It is also a good idea to mix each sample prior to placing on the transfer plate. You will take 20 microliters of each patient sample and place in the corresponding well on the transfer plate. The controls are treated in the exact same manner as the patient samples, meaning that 20 microliters from each control will go into the corresponding well. Now that we have diluted all of our patient samples and the controls in our transfer plate at a 1 to 10 dilution, we will now take 50 microliters of each of these patient samples and transfer into a corresponding well on the transfer plate for a final 1 to 50 dilution. A pipette that has the multi-channels is much more efficient in this process. It's a good idea to go up and down a few times in the well to make sure that it is properly mixed. We will now move this transfer plate off to the side for a few minutes while we make our standard curve dilutions. To make the standard curve dilutions, you will want to label each one of these test tubes with a corresponding concentration. I have already done so. You will now use your standard diluent and place 400 microliters from the standard diluent into each one of your test tubes. Gently vortex the kit calibrator and look to make sure that it has all been reconstituted. Next, you will transfer 400 microliters from the calibrator vial into your 200 nanogram per ml sample. Since this is a serial dilution, it is necessary to mix each tube of sample. You will then take 400 microliters from this sample and place it in the next vial. Now that you have completed the standard curve dilutions, it is a good idea to transfer them onto your transfer plate. This ensures a more rapid transfer onto the actual antibody-coated plate. A volume of 250 microliters per standard curve dilution is an appropriate volume. Now that we have our transfer plate ready to go, we're ready to begin the actual assay procedure. We are now ready to transfer our patient samples and the standard curve onto our antibody-coated wells. You will notice that the wells are removable, so if you are not running an entire plate, you can simply pop them out by flipping the plate over and pushing down. You will want to then take these rows and place them back into the bag with the desiccant pack. Seal it and place this back into the refrigerator until you are ready to use it again. However, we are going to be running an entire plate and will need all of the rows. Set your timer for 1 hour, but do not press start until you have transferred the last of the patient samples. Be transferring 100 microliters in duplicate from every row onto the antibody-coated wells. Be careful not to scratch the bottom of the wells with your pipette tips. Use of the multi-channel pipette for transfer is critical because the transfer must occur within 5 minutes. Press begin on your timer and move your plate to a plate shaker and shake for 1 hour at 750 RPM. During the first incubation period is a good time to get your wash station all set up. Our plate washer uses a special bottle to pull the solution from. You will have to check your individual requirements and transfer the wash buffer into the appropriate container. At the end of your first 1 hour incubation, place your plate into the plate washer and press start. Once the plate washer program has finished, check to make sure that there is no remaining residue inside of the wells. If there is, simply tap the plate on some lab paper. You can now proceed to the next step of adding the biotinylated antibody to each of the wells. The next step in the assay process will be to add the blue biotinylated antibody solution to each of the wells. To do this rapidly, pour your solution into a reagent reservoir. Next, you will want to use a multi-channel pipette and transfer 100 microliters of the solution into each of the wells. Set your timer for 1 hour and place the plate on a plate shaker, again at 750 RPM. After the 1-hour incubation with the biotinylated antibody, repeat the wash procedure that you previously performed. Place your plate in the plate washer and begin the program. The next step in the assay procedure is to use the streptavidin reagent that we made at the beginning during the setup. Transfer this solution into a reagent reservoir. Then transfer 100 microliters of the solution into each of the wells using a multi-channel pipette. This incubation time is 30 minutes. Make sure to set your timer for 30 minutes. Place the plate on the plate shaker at 750 RPM for the remaining of the incubation time. After the 30-minute incubation with a streptavidin reagent, again place your plate into the plate washer and begin the wash procedure. The next step in the ST2 assay procedure is to add 100 microliters of TMB reagent into each of the wells. Please note that TMB is highly reactive. You do not want it to come into contact with metal. When you pour it into your reagent reservoir for the transfer, make sure that the solution is clear. If it has any color whatsoever, it should be discarded and the assay will have to be rerun on a new kit. Transfer 100 microliters of TMB reagent using a multi-channel pipette. Set your timer for 20 minutes and begin incubation again on a plate shaker, but this time ensure that the plate is in the dark. This can be accomplished by placing your plate shaker in an incubator with the door closed, or simply by placing any sort of cover on top of it. Again, the incubation must be done at 750 RPMs. The final step of the assay procedure is to add 100 microliters of stop solution into each well. This solution will go on top of the TMB that is already in the wells. You will notice a color change in the solution from blue to yellow. Now gently tap the plate to make sure that the reaction has completely stopped with the stop solution and TMB mixing. Do a visual check to make sure that there are no air bubbles in any of the wells, as this could affect the OD reading in the plate reader. After you have added the stop solution to each of the patient wells, you may now place them in the plate reader to obtain OD values. Refer to your owner's manual and the instructions for use on the assay for the requirements of the plate reader. Depending on the configuration of your plate reader, it may or may not provide you with the ST2 concentrations. It might only provide you with the OD values, in which case please refer to the following section on analyzing results for further instructions. Thank you very much for watching our training video.